I'm very happy to introduce our next speaker, who's also a returning ARDD star. I think you've been
10 times.
10 times. You've been here 10 times. Amazing. Alexey, whenever you're ready, Alexey Moskalev, take it away.
Dear colleagues, I'm really happy to be here, and it's my 10th ARDD conference. Thank you, Morten, thank you, Alex and Gary, for this opportunity, and I would like to discuss with you the scientific and clinical application of biological age. You know that biomarkers of aging are parameters that including molecular, cellular, physiological, and psychological aspects that correlates with the chronological age and also predicts mortality and disease of aging. And it's not aimed only to predict these events, but also to look at the effectiveness of interventions in clinical studies. And we could use it as endpoints in our clinical studies to invent new aging preventive or aging reversive interventions. The key criteria of biomarker of aging, from my side of view, to be sensitive to early signs of aging that are distinct from mortality or frailty index, to have a predictive function within a reasonable time range, and have low analytical variability, high robustness, and reproducibility. But the problem is that a singular indicator as an aging biomarker not meets all the specified criteria, and each biomarker of aging has both advantages and limitations. It is worth considering panels of biomarkers that reflect different aspects of aging, or a complex biomarker based on them, so-called biological age or aging clock. Several years ago, I proposed a classification, very simple classification of biomarkers of aging, divided them into 4 groups. First 2 groups, although these markers, like clinical and functional, possess clinical significance, the ability to predict mortality according to chronological age remains uncertain in terms of the extent which they reflect biological aging itself, rather than health issues induced by other factors. Furthermore, these markers commonly function as aggregated indicators in big samples, in big cohorts of individuals, but exhibit a substantial variability among individual measurements. The third group is molecular group, which corresponded to mechanisms of aging, specific mechanisms. For example, you could measure stiffness of extracellular matrix, or cellular senescence, or telomere length. Of course, it's more vulnerable to early stages of aging at the preclinical stages, but still have individual variability because aging is a complex process involving multiple levels of interaction, and the markers targeting specific mechanisms cannot capture all the aspects. And the more promising could be omics biomarkers, including such kind of omics like transcriptome, methylome, proteome, metabolome, metagenome. These omics approaches allow us to examine changes in the organism at different levels, providing valuable insights into the complexity of the aging process. And additionally, we could use a multi-omics approach, combining different omics. very recently will be published a paper in Cell from a big consortium, Biomarkers of Aging Consortium. This consortium was organized by Vadim Gladyshev and Mahdi Mokri. And the aim of this consortium to develop a framework for classifying and evaluating biomarkers that can be used to identify and assess interventions that could promote healthy longevity. This involves a consensus on terminology, regulatory classification biomarkers, illustrating use cases based on existing biomarkers in trials, and clinically evaluating biomarkers using preclinically validated geroprotectors. It could be published maybe today or tomorrow, this paper. Many years ago, Alex Zhavrankov and our team provided the first AI-based blood clock of aging, consists of dozens of parameters of our health, measure it using biochemistry and hematological approach. This clock is quite accurate and simple for using. You could go to aging.ai and put your parameters and calculate your blood age using this. And recently we applied this aging.ai 3-version clock to our clinical data obtained from the medical spa clinic that are applying 2 weeks of treatment, preventive medicine approach using healthy diet, caloric restriction, physical training, exercises, and also some supplements and medical spa procedures. And after 2 weeks, we could see that in most cases we observed a decline of this parameter. In this point of view, younger individuals after 2 weeks of treatment, up to 3 years of biological age using aging AI. We did also so-called artery indices. We used machine learning to see the predictivity of 68 different parameters of health in 200 individuals of different ages, healthy individuals, and did rating of these parameters and observed that parameters of artery health are more predictable than other kind of parameter, including endocrinology, inflammatory, etc. And we did a clock based on 4 parameters of cardiovascular health using ultrasound of carotid arteria and applanation tonometry of arterial stiffness in one model. We observed that patients with hypertonia and diabetes have higher biological age using artery indexes. And recently we also did a prospective randomized comparative placebo-controlled double-blind study in 2 groups to assess the effect of the complex supplement from terpenes in combination with the linen oil to study these artery indexes, and observed that this parameter after 3 months of application dropped by 2.5 years compared to the baseline. And in the control group, in placebo control group, we could not see any statistical significant changes. The pulse wave velocity significantly decreased compared to the initial one by 10%, and also the minimum thickness of intima media complex of carotid arteria is also decreased after the treatment. In the COVID period, we tried to apply different blood aging clocks to patients entered to the clinic with the COVID and compared the biological age correlation with the severity or mortality of these patients in the first year of COVID. And we used 6 kind of blood clocks in this experiment, and I presented only 4 on this picture, and we could see that Climera and Duval biological age and FENA age are very accurate in prediction the severity and mortality of that COVID patients at the starting of treatment in the clinic. In collaboration with my friend who accidentally died from COVID a couple years ago, Arnold Mitninsky, we did estimation of frailty index in that patients, and observed that this complex parameter was invented by Arnold Mitninsky and co-authors many years ago. In survivors, twice less than in patients who died from COVID. This is very powerful predictor of mortality, better than age and sex in these patients. And yesterday I received from my staff member this slide. We did another clock, echo age, using echocardiography parameters of healthy individuals of different ages. And we could see that this kind of aging clock are quite accurate in uninvasive, because you use only ultrasound technique for your head, and could see many parameters that after putting into this model predict your biological age. This is my biological age, big triangle. I'm 46, but by this model I'm 31. But we also applied this clock to 4 young patients with Huntington-Gilford progeria, children with a chronological age from 4 to 8 years old, but their functional biological age using EHA you could see range since 46 up to 60 years old. I hope it could be helpful as a clinical biomarker in clinical studies. I'm administrator, for now I'm administrator a big group of bioinformaticians that are using explainable artificial intelligence. It is kind of modeling with such advantages as increased confidence in the model, detects early problems with the model generalization, and contestability. We could know which parameter could affect the results more than other parameters. And my colleagues applied this approach to their experimental data based on 38 or 10 cytokines of inflammation. And they did 2 kind of inflammatory clocks in collaboration with Claudio Franceschi. The small inflammatory clock were published very recently, last month. and it's quite accurate and predicts severity of different diseases, including rheumatoid arthritis and cardiovascular risk. And they also did cognitive clocks, not by psychological measurements, but by psychophysiological measurements, including sensomotor parameters and compimetry, discrimination of color on the screen of the computer. And also combine this with the blood biochemistry and DNA methylation clocks, and provided very accurate clock for our cognition. As a conclusion, I hope that the modern medicine that proposes aging as a normal physiological state that does not require intervention and treat symptomatically many aging-related diseases will shift into longevity medicine, where aging is a disease-like state that needs to be treated, and we will do as much as possible clinical studies using aging clocks and eliminate the causes of chronic diseases because of these clinical interventions, and prevent age-related diseases which could allow us to reach healthy longevity. Also, I would like to bring your attention to our books, where I was, or as an editor or as a contributor, that could be helpful for clinicians, for example, for scientists as well, to do clinical application of different biological clocks. And my colleagues did GPT-like Telegram bot. On the corner of this slide, you could use this QR code to access. And it used our publications, also our, popular publications about health prevention and anti-aging, and ask this chat about aging. Thank you for your attention.
Thank you so much, Alexey. We have time for a question if someone wants to ask.
Didier?
Yes, thank you very much, Alex. Very interesting. In one slide, you wrote using machine learning algorithms, we have developed a model that accurately predicts chronological age based on 4 non-invasive diseases. Can you develop a bit which algorithm you use? Is that, let's say, between brackets, open source? Is that available?
Yeah. Do you mean this slide? Yes. It was published in Aging in 2017, and I could share you the paper and we could discuss. If you have any medical data, we could do it, new models with you using XI, Explainable AI. I would be happy to collaborate.
All right, thank you so much, Alexey. Fantastic talk.